Okay, hello to you all. My name is Noma Assad and I am doing a PhD. I'm in the first year of my thesis now in the team of Professor Mabaza. So my project is more translational research more than it's clinical, concentrating on dipeptidyl peptidase 3, the source and the mechanism of release in pathological situations. So to start with, DPP3 is a metallopeptidase which cleaves dipeptides sequentially from the N-terminus. It has several substrates such as angiotensin, enkephalin, and endomorphin. It is intracellular, mainly cytosolic, but it is also detected extracellularly in body fluids and in the plasma of healthy and sick individuals. So clinical data show that increased levels of circulating DPP3 were associated with increased mortality risk in patients with cardiogenic shock, hemorrhagic shock, and septic shock, showing DPP3 as a biomarker of prognostic interest. Moving to preclinical sets, actually injecting healthy mice with human DPP3 led to a significant decrease in left ventricular shortening fraction, showing DPP3 as a myocardial depressant factor. So as you see here, DPP3 is ubiquitously expressed in all of the cells and the tissue of the body. And we see that its expression is differential based on the type of cell. Here we are speaking in physiological situations. However, in pathological situations, it's not yet understood or not well documented the expression of DPP3. So from here comes the objective of our study, to first detect the DPP3 expression in pathological situation, second, detect the source of this protein, and then third, how it is liberated from tissues into the circulation. We have several hypotheses regarding the source and regarding the release mechanisms, cell lysis, exosomes, shedases enzyme, but I will concentrate today on the first objective, which is the DPP3 expression in pathological situations. So to do so, we used two animal models, two validated cardiac dysfunction model. The first is by injecting isoproteinol 300 milligram per kilogram for 2 days, 2 times per day, and then the third day we sacrifice the animals. And doxorubicin model, 4 milligram per kilogram per day for 6 days, and then after 1 week we sacrifice. So at the end of both of the models, we harvest the organs, the blood for obtaining plasma and leukocytes, and bone marrow for obtaining bone marrow cell pellet and the supernatant, the stroma in this microenvironment. And then we extract proteins and we extract mRNA and we study the expression of DPP3 on protein level and on the mRNA level. So our results was, here we see the results in the isoproteinol model. In each organ we have mRNA and protein expression. We will concentrate more on the protein in analyzing the results. We see here in these 4 organs, the heart, liver, kidney, and lung, we see the tendency to decrease or to increase, but no significant results. And it's always different between the different organs. Now, what we have seen a significant change is in the spleen. We've seen a decrease in the expression of this protein in the iso group of mice compared to the control group. What was quite interesting for us is the increase in bone marrow cells and in the bone marrow supernatant surrounding these cells in this microenvironment. Now moving to the doxorubicin model, we have here the results on the protein level. We have seen that there was a decrease, significant decrease in the heart and the lung in the group of doxorubicin mice compared to the control. No significant changes in liver and kidney. In the spleen, we see increased expression of DPP3, and in the bone marrow cells and supernatant, we see like a tendency to increase, but it's not significant. So actually here, by that, we detected the expression of DPP3 in 2 different animal models. And if we want to analyze these results and see how they can orient us in our next objective, which is detecting the source of this protein, we have actually 3 hypotheses. So first of all, either the tissue or the organ that highly express DPP3 in this certain pathological situation, it is the organ that liberates DPP3 into the circulation, which in this case scenario we have in the iso model bone marrow cells as a like a candidate, let's say, for releasing DPP3, and in the doxo model we have the spleen. Or the tissue that we see that it's a decreased expression of the protein means that it already released the protein into the circulation, which leaves us with the spleen in the iso model and the heart and the lung in the doxo model. Or there is no correlation between the expression and the release into the circulation, which leaves us with all organs actually. Now we are currently in process of continuing additional experiments. We are analyzing the activity of DPP3 in plasma samples and in bone marrow supernatant by collaboration with 14-4 company, and we are studying also the localization of DPP3 by immunofluorescence. So as we have seen, we've seen different expression of DPP3 among the two models. So it is maybe due to the fact that it's expressed differently based on the mechanism of each pathology or mechanism of the model. For example, in isoproteinol model, we see ischemia, we see hypoxia, necrosis, we see oxidative stress. In doxorubicin model, we have also oxidative stress, but we see more cardiomyocyte damage, endothelial dysfunction, mitochondrial dysfunction, and apoptosis. Plus, the fact of the systemic implication in each model. For example, in the ISO, the heart is the main target, while in the doxorubicin, there is more systemic implication as a manifestation. Second, the different time points between the two models, the duration of the model and the time we sacrifice the animals, because for us, the validation of the model is always based on the cardiac function, but for the modification on the tissue or on the molecular level, it doesn't have to follow the same time pattern of the cardiac function, actually. So now we have more specific experiments in the next step to know exactly the source of DPP3. Just I have a couple of words regarding the mechanism of liberation. We have just a very preliminary start. As studies show that in pathological situations, DPP3, the increase of its level is associated with increase of cardiac and heart cell lysis markers. So what we have started to do to validate or to examine this hypothesis that DPP3 is liberated by cell mortality or cell lysis, we started in vitro experiments on cardiomyocytes, and we stressed the cells. And we have seen that the more we stress the cells, the more we starvate them, the more we see mortality and the more we detect DPP3 in the supernatant of the cells. And we are currently now trying different stress conditions, hypoxia, doxorubicin treatment, etc. And the next, it will be working on animal model and cohorts and trying to measure DPP3 in plasma, in tissue, cell lysis markers, and mortality rates, and try to find if there's a correlation between these parameters to know if it's related by cell lysis. And thank you for your attention.
Okay, do we have any questions? Or do I get to ask them all? Apparently me. Okay. So I'm sitting here and Alex is whispering to me that this is going to be a really big marker someday. Yeah. So as an ER doc, I have some questions about things that are going to be markers, because the first thing is I want to know how stable is it when I draw it out of the patient? Does it fall apart in 2 minutes and I have to put it on ice and get it analyzed quickly, or can I set it on the table for an hour? Is it, because my experience is most of these kind of things don't last very long in the serum.
For me, when I worked on it actually in vitro, when I treated the cells, because I had previous experiments, we worked on ice. I know the half-life in the body, but actually I don't have a specific idea of it. For me, I work on ice, but I don't know the degradation of DPP3 specifically. I don't have an idea of this information actually.
Do you have a feel for the half-life in the body?
It's 20 minutes.
Okay.
Yeah.
And it's eaten by the blood?
It's actually, when we were trying to put the hypothesis regarding this project, we had also another axe in our mind, which is the degradation or clearance from the body. So we don't have specific now data regarding this, if it's the liver, if it's the kidney, if it's both, if it's...
Okay. And the last question I have is, what are the measurement units? Is this a milligram per deciliter, a nanogram per liter? What is the endogenous concentrations that you're working with?
Actually, in the clinical data, it was nanogram per milliliter.
Okay.
For a normal in the plasma, and for the activity we measure the enzymatic activity. Yeah.
So it might be an ER candidate one day. All right. Anybody else? Yes, back here.
Thank you for this very good presentation. I'm not a troponin expert, but I'll just try to imitate the guys that do all the troponin work. And you showed that an elevation of DPP3 is associated with an elevation in cardiac troponin I. And you also said that the isoform was a more cardiac-specific injury, perhaps in docs it was a more systemic inflammation or systemic reaction. So do you have any information about if an elevation in DPP3 is also associated with an elevation in troponin T? And if perhaps in the docs variant there's even more association with troponin T compared to troponin I?
Actually, we didn't measure yet the troponin in the samples. It was also one of the steps that we wanted to do next. In these two animals models in the samples, in addition to measuring DPP3 activity in the plasma, we wanted to measure troponin, but we don't have yet data regarding the increase of the troponin.
Okay, thank you.
Thank you.